Hi, I'm Dr. Zoe Waller and I'm an Associate Professor in Chemical Biology in the School of Pharmacy at UEA. The aim of this video is to explain our recent research paper to a general audience. It's just been published in Nucleic Acids Research, which is an open access journal. This means that anyone can read it, you don't need a subscription. The links to the paper are included on the YouTube webpage. The title of the paper is Epigenetic Modification of Cytosines fine-tunes the stability of i-Motif DNA. The work is the result of a collaboration between my group in the School of Pharmacy at UEA and Dr. Nicole Smith's at the University of Western Australia. So what's the paper based on? Well, our bodies are made up of many different types of cells. Within each cell is a nucleus, and this is where our genetic information is held in the form of DNA. DNA is made up of 4 building blocks which we call bases, A, T, C and G, otherwise known as adenine, thymine, cytosine and guanine. In DNA, 2 strands of the building blocks coil together to form what is known as a double helix. This structure is a bit like a twisted ladder where the links between the strands, or the rungs of the ladder, hold them together. DNA, which is composed mainly of the bases G and C, can form unusual structures such as G quadruplexes and i-Motifs. These look very different to the typical double helical structure, which was first proposed by Watson and Crick in 1953. My group are interested in i-Motifs, which form in DNA which contains lots of the base cytosine. In the genome, the base cytosine is also used like a bookmark on the DNA. Chemical tags mark our DNA in response to changes in the environment. These tags can turn genes on or off. As the structure that we're interested in is composed of cytosines, we considered that it was important to see the effect these chemical markers have on their structure. So what did we already know about i-Motifs? Well, we know that in biology, i-Motif structures can affect how genes are read. For example, normal gene expression starts in a position called the transcription start site. We know that alternative DNA structures can alter how the gene is expressed when they form in front of this particular region. This can determine essentially whether the gene is switched on or off. But not all genes have these sequences, and not all sequences will form i-Motif under physiological conditions. Some need acidic conditions to form, some do not. But the DNA-based cytosine is actually used as a marker in DNA. This also determines whether genes are switched on or off. There are 4 different types of markers that can be added to the base cytosine, and these are called methyl, hydroxymethyl, formal or carboxyl, and I've depicted them here as hats on the cytosine. These all have different effects in how genes are read. Some work had already been done to look at how these affect i-Motif DNA, but we wanted to do something a bit more comprehensive. So what have we found? Well, we looked at an i-Motif forming DNA sequence and then added the special cytosine markers. We looked at the effect of each position where there is a cytosine in this sequence, one by one, carefully replacing the cytosine with a tagged cytosine. We did this for each type of marker, and if you're counting, there are 12 cytosines in this sequence. Doing this with 4 different markers gave us 48 separate sequences which we looked at, and that's on top of the unmodified DNA. Well, what did we find? We found that there were particular parts of the sequence which were most affected by these markers, and these actually formed in a very, very close proximity to each other when they were folded in the i-Motif shape. We then looked at the DNA from the human genome to see where these markers overlap with known i-Motif forming sequences. We also looked at the markers in cancerous and non-cancerous cells to compare, and we found that the majority of sequences were bookmarked differently in cancerous compared to non-cancerous cell lines. i-Motif forming sequences which were known to be stable at neutral pH were also significantly more likely to be bookmarked or tagged than those that were known to be unstable. But what does this mean? Well, the work hints at a relationship between i-Motif DNA structures and the markers on DNA and how they are used for whether genes can be switched on or off. It also indicates that these markers are different in normal and cancer cells. Our understanding of how these markers affect i-Motif DNA structures can also help us use them better when we are using these structures in designed devices which uses DNA as a material. This work has been supported by the BBSRC, or the Biotechnological and Biological Sciences Research Council, and the Eastern Academic Research Consortium. If you have any questions, please feel free to ask either by tweeting me, adding a comment, or by email. Thank you very much for watching, and bye-bye for now.